So here we are going to first define what non-competitive inhibition is, and then in a later video we are going to go over the Michaelis-Menten and Lineweaver-Burke plot to further define this type of inhibition. Now, before jumping into what non-competitive inhibition is, we have to remember that when we talk about inhibitors for enzymes, there are two major classes of inhibitors. We have irreversible inhibitors and we have reversible inhibitors. Now, irreversible inhibitors are able to form covalent and non-covalent interactions with our enzyme, and they are irreversible. Whereas reversible enzyme inhibitors only form non-covalent interactions. They do not form covalent interactions with the enzyme. So non-competitive inhibition is an example of reversible enzyme inhibition. So it forms non-covalent interactions. Now, non-competitive inhibitors bind to the allosteric site of the enzyme. Now, if we take a look at an enzyme, remember we have two locations of interest. We have the active site and then we have the allosteric site. Now, the active site is where the substrate will come in and bind and the enzyme will convert that substrate into product. But when we talk about non-competitive inhibition, the non-competitive inhibitor will not bind to the active site. Instead, it is going to bind to another site, which is called the allosteric site. Now, if we compare this to competitive inhibition, remember that in competitive inhibition, the inhibitor is binding to the active site. But in non-competitive inhibition, we are binding to the allosteric site. Now, another thing to understand of non-competitive inhibition is that at what point does it bind to the enzyme? Remember that when we talk about the catalysis of a reaction with an enzyme, we start off with enzyme and substrate. The substrate will come in and bind to the active site of the enzyme through the induced fit model. So we get this ES complex, enzyme substrate complex. And then our enzyme substrate complex is altered into our enzyme product complex because the enzyme will convert that substrate into product, and then the product will leave the active site. And that is our general catalysis reaction. At which point in this reaction will a non-competitive inhibitor bind? So a non-competitive inhibitor can bind to free enzyme, or it can bind to the enzyme substrate complex. So it can bind independently to either or of these situations to form an enzyme inhibitor complex or an enzyme inhibitor substrate complex. And this is really important to understand, especially when we go on to learn about uncompetitive inhibition. And that is essentially non-competitive inhibition. We will go over the Michaelis-Menten and Lineweaver in the next following videos. Just one thing to note before going into the next videos is that for non-competitive inhibition, our Vmax, so our maximum velocity, will decrease, whereas our Km, our substrate concentration at 1/2 Vmax, will remain the same.